Boa tarde a todos. Mais uma vez, com muito prazer e muita satisfação, damos início aqui aos nossos ciclos de webinários, versando sobre temas voltados aí tanto para a biologia molecular quanto para a citogenética. E hoje contamos dentro desse contexto de palestras voltadas aí para ferramentas interessantes à biologia molecular, principalmente voltados a reagentes específicos dentro de fluxos de trabalho nos laboratórios. Hoje contamos também com a presença da doutora Emily Putnam. A doutora Emily é especialista, responsável por desenvolvimento de produtos da empresa QUANTABIO, uma parceira da Citogem. E hoje ela especificamente vai conversar um pouquinho sobre uma linha de reagentes especificamente voltadas para o preparo de bibliotecas, voltados aí para reações e fluxos de trabalho em NGS. Então ela vai mostrar um pouquinho como funciona esse material, quais são as suas características, como é a sua performance. E eu convido a todos então para que possamos ouvi-la e possamos aprender um pouquinho sobre essa linha interessante de reagentes de biologia molecular. Aqui também presente, conto com a presença de meu colega Dr. Alexandre como mediador deste webinar e espero que seja bastante proveitoso a todos. E no final da palestra podemos aí abrir espaço para questionamentos e perguntas. Emily, so we are ready to get started. Thank you very much for joining us in this webinar. It's a pleasure to have you with us as well, and you are good to go. Thank you very much again.
Great, thank you so much for that lovely introduction. So today I will be discussing the Spark NGS library preparation kits from QUANTABIO, and we'll be discussing the options that we have for both DNA and RNA sequencing. Just as a brief background for those of you who may be unfamiliar with QUANTABIO, we were formed in 2001 by former amplification scientists, and we really specialize in engineering application-specific enzymes and optimized reagent composition with our popular tough mix additive for superior product performance. As you can see here, we were formed over 20 years ago, and throughout the years we have introduced a variety of products that have been used for everything from cDNA, qPCR, as well as RT-qPCR technologies. And in the last few years, we have leveraged the enzyme expertise that we have to generate new library preparation kits that will be faster and easier than other kits you may be familiar with. In just the past few years, we have also introduced several options for RNA sequencing as well, and that will be the focus of today's talk. So again, we have products throughout our portfolio for all of the options seen here. For other reference for these other products, we do have other webinars that we have done in the past, which should be available on YouTube. But today I will be focusing on next generation sequencing. So the typical next generation sequencing workflow starts with library preparation. From there, it goes directly on to this could be an Illumina instrument. This could be several other sequencing instruments that are available on the market. But this is the traditional overall workflow. And the key step in the process is this library preparation, because whatever you put into the instrument will be directly related to how good the data quality comes out. So for us, it's very important that our library preparation kits are designed to not only be fast and easy, but also to generate good, high-quality libraries so that you get good, high-quality data. This is an overview of our Spark NGS portfolio. I will be going through the various options presented here, but we have everything from DNA sequencing through RNA sequencing and the supporting products to help you make fast and easy library preparation. Altogether, we have a very complete DNA and RNA library prep portfolio so that you may tackle several different workflows in your labs. Starting with DNA, I'll be focusing on our Spark Fragging Library Prep Kit and our Spark DNA Library Prep Kit. So altogether, these are a very easy kit for DNA library preparation. specifically for whole genome sequencing, shotgun metagenomics, anything where you require high quality DNA for library prep. And we have two options here. The workflows are very similar. So starting with our Spark DNA library prep kit, this takes as input fragmented DNA from down to 250 picograms input all the way up to 1 microgram. This fragmented DNA can be anything from FFPE to cell-free DNA to amplicons of a small size. Basically anything that is already suitable for short-read sequencing would go directly into the Spark DNA library prep kit. This first tube only involves DNA polishing and end repair, which enables adapter ligation to take place directly after. The other option that we offer is our Spark DNA Frag and Library Prep Kit. And the difference here is that this module includes a fragmentation enzyme so that you can take intact, high quality, high molecular weight DNA and fragment it down to the correct size for sequencing. In this case, the input ranges from 1 nanogram to 1 microgram, and this first step is tunable depending on input amount and desired fragment size. Both kits at this point go directly into adapter ligation and cleanup, and this is done in the same tube. So there's no additional manipulation here, no additional cleanup between the first step and the adapter ligation. And this is going to make it much easier from a handling perspective as well as from a time perspective. After adapter ligation is completed, we have the ability to safely stop here. And there's also the ability to do optional library amplification. So for anybody who may be working with at least 100 nanograms starting input DNA, if you choose, you can actually entirely skip the PCR amplification, and your libraries would already be ready to go after adapter ligation. Alternatively, if you're working with very limited sample inputs, you should use the optional library amplification module, and this will be just an additional 50 minutes after that bead cleanup. There'll be a second bead cleanup after library amplification, and the supernatant will go into the third tube, which will be your sequencing ready library. The overall turnaround time for the workflow is about 2.5 hours, and the hands-on time is very limited, with only about 30 minutes required for hands-on time. Overall, as you can see, this is a very simple workflow, making it easy for both manual library prep as well as for automated library prep as well. A key feature of our kits is the ability to have very uniform coverage across different GC contents. Very popular methods on the market often include transposase-based methods, which make this very fast and easy to use. But as you can see with the yellow line here, there is a very strong bias towards the middle GC content range. And especially for whole genome sequencing, it's imperative to be able to cover the whole genome uniformly at both high and low GC contents here. And you can see, ideally, we should have a nice straight line around 1 for normalized coverage, indicating that we have uniform coverage across the length of the genomes. Here we see in orange the Spark DNA Frag and Library Prep Kit, which is very close to that approximately 1 normalized coverage. As a comparison, so we also have in black, this is Kavara's sheared sample that's gone into our Spark DNA library prep, and we see good concordance there as well. So altogether, these two kits minimize bias and maintain very even coverage across a broad range of GC contents, enabling this kit to work well with not only human or other similar mammalian samples, but other also bacteria, plants, other samples that may have differing GC contents will also be well represented with these library prep kits as well. Another key feature is that, again, leveraging our enzyme technology, is the ability to have a very high library prep efficiency. So what we are looking at here is the correlation between the input amount and the resulting DNA library. So what's most important is that even at very low input levels, all the way down to 100 picograms, what we see here is that what you put in is what you get out, indicating that there is very superior library efficiency, especially with the ligation. And it means that you're going to get superior data coverage at the end of your library prep, regardless of the starting input amount. Another key question that we often see is whether the hi-fi polymerase that is included with our library prep kits is introducing bias. This has been a key factor in library prep historically, but again, with our technologies and our hi-fi enzyme, we don't see that amplification bias. So here on the left, what we've done is we've taken a mixture of 3 bacterial species at different GC contents, and you can see that in the dark gray. And what we've done is we've compared an amplified library in orange with a PCR-free library in blue. Again, we should see a nice straight line around 1 for that normalized coverage, and we see that indicating good uniform coverage across the genomes, as well as very high concordance between the amplified library and the PCR-free library. And this indicates that the hi-fi amplification is indeed high fidelity and that the sequence fragments are true to the genome being represented. Of course, everybody is always concerned about the library yields as well. And we can see here on the right, looking at our Spark Hi-Fi PCR master mix, which is included with the library prep, that regardless of input amounts, we always see higher yields, and again, indicating that you can use reduced cycling to achieve sufficient library yield without introducing bias. Finally, we also take a look at duplication reads. This is also an indicator of PCR bias or over-amplification. And as we can see here, the duplication reads for both these mixed genomes as well as single genomes is very, very close from the amplified library and the PCR-free library, indicating that you do not need to be concerned about the additional library amplification steps. Now, at this point, I would like to switch to a discussion of our RNA-seq library prep kits. Now, RNA-seq is a little bit different and requires a little bit more consideration in terms of the choice of the library prep kit. And this is inherent to the RNA composition of the cell. So here we're looking at overall RNA composition. And as you can see, over 80% of the ribosomal or of the RNA content is ribosomal RNA, which is not interesting for gene expression analysis. This is highly repetitive RNA. And what this ultimately means is if ribosomal RNA is present in your library, you're having wasted reads, you've spent a lot of additional money for sequencing to get what you're interested in. We also see about 15% is tRNA, which again is not interesting for gene expression analysis. And the remainder is 3% mRNA, and then about 2% other subtypes, including long non-coding RNA, microRNA, etc. So altogether, at the very most, only about 5% of the cellular content is interesting for gene expression analysis and interesting for library preparation. So this makes it especially critical for choosing a library preparation kit that focuses only on the things that you want and doesn't include that ribosomal RNA, which is not interesting for gene expression analysis. There are 3 main methods for RNA library preparation. The first is poly-A selection, which utilizes the fact that high-quality, intact mRNAs from mammalian and plant samples have this intact poly-A tail, and they use that to select specifically for those mRNAs for library preparation. Another method is rRNA depletion and globin mRNA depletion, which depletes out everything that you're not interested in, leaving behind only the subtypes that are interesting for analysis. I'll be talking about both of these options today. I just briefly also want to mention there is a third method of size selection. We do not currently have anything for that method. But the poly-A selection and the depletion-based methods will cover the vast majority of RNA transcriptome analyses. So now I'd like to introduce the first of the RNA-seq kits, and this will be our Spark RNA-seq HMR kit, and this will be for whole transcriptome analysis from human, mouse, and rat samples. So when we say HMR, it's a reference to the sample type that is specific for this kit. This utilizes ribosomal and globin mRNA depletion so that, again, the RNA that is not interesting is depleted out, and you're left with long non-coding RNA, those interesting subtypes, as well as your mRNA. Depletion-based methods are also the preferred method for any sort of degraded sample type, such as those from FFPE, where the poly-A tail may not be fully intact. So overall, we have made this an incredibly simple and efficient workflow. Historically, RNA-seq can take up to 8 or more hours. There can be 20 plus components, many steps, and can be overall a very complicated and hard to use method. And we've taken that and we've made this significantly simpler and easier to use. So overall, we have 3 reaction tubes, a total of 9 steps and 10 components for this efficient depletion of globin mRNA and ribosomal RNA for human, mouse, and rat samples. The input for this will be total RNA. This can be from blood or any other sample type. And the input ranges from 1 nanogram up to 1 microgram. This kit is also, again, as I mentioned, compatible with FFPE RNA. For that, we recommend between 10 nanograms and 100 nanograms, and that is to account for the fact that FFPE RNA is very degraded and difficult to work with. From there, you then can start the library prep going into the first tube for the first set of reactions. So what you can see here in tube 1 is that we've broken this down into a series of sequential reactions where you simply add in the additional buffers and enzymes needed to complete the next step. The first step in tube 1 will be fragmentation down to the appropriate size for short-read sequencing, and will also include that ribosomal RNA and globin depletion step, as well as random priming. Directly into that same tube, after that reaction has completed, you'll then add in the next set of reagents for first-strand cDNA synthesis. Again, once that's completed into the same tube, you'll then add in the third set of reagents for that second-strand synthesis, DU strand marking, and finally end polishing. After that, you then go into your first bead cleanup, which features our novel technology, which is our bead booster buffer. The bead booster buffer is included with the library prep kit and will be added to the magnetic beads for cleanup. This bead booster, again, is novel for our technology, and it makes for easier and more efficient manipulation of the beads for better cleanup and higher yields. After that first bead cleanup step, you then go into adapter ligation in tube 2. And again, this is a very short ligation step, which features any sort of UGI Y-shaped adapters, such as our Spark UDIs. Then you go into a second bead cleanup, again using that bead booster for easier handling and more efficient yields. Finally, you then go into tube 3 for the final reaction, and this features strand-specific PCR enrichment, which enables strand-specific information from your library. You'll go into a final set of bead cleanups, and then you will have your double-stranded library ready to go. And this total time is about 4.5 hours. This also features about 30% less hands-on time than other kits on the market, making this incredibly fast and easy to use. Of course, anytime you're working with a depletion-based method, it is especially critical to ensure that you're getting efficient removal of both the ribosomal RNA as well as globin mRNAs. I have decided just today to share some FFPE RNA data. Since FFPE is more difficult to work with, this showcases how efficient our enzymes are in particular. Just for reference, the FFPE RNA that's being worked with here has a RIN score of around 3.5, so pretty highly degraded sample. And what we can see here on the left is looking at high input, what we consider high input FFPE RNA at 100 nanograms. And what we see here is very minimal ribosomal RNA in that dark blue, leaving us a very, very good amounts of sequencing reads, especially for exonic regions, which are shown in green, roughly 80%. And you can see in comparison to other kits on this market, this is more highly representative of the sample breakdown that we would expect to see for these subtypes. Again, even with low input, so we've tested here 25 nanograms and 10 nanograms, again, of that FFPE of low quality, we still see very consistent ribosomal depletion, around 1% or less, as well as over 80% of the reads aligning to exonic regions, meaning that we get very, very high quality DNA reads and nice mapping rates to the areas that are going to be most important for analysis. And as we can see here, even with degraded sample types, we see very consistent depletion regardless of input levels. Moving on, as I mentioned, we also focused on globin mRNA depletion, which allows this kit to be used with whole blood samples as well. Here we've looked at both high and low input whole blood RNA. And we see, again, very consistent depletion as we would expect for the ribosomal RNA, which is shown on the left. And we also see very, very high depletion of those globin mRNAs that are going to be present in high amounts in whole blood samples. So even here, this shows the compatibility with whole blood samples. And again, very, very high quality results for efficient analysis. Here we're looking at improved library yield. Looking on the left, what we're comparing is UHR RNA, which is a high quality universal human reference RNA, and we've compared this against some of our main competitors on the market, and we've compared them at their lowest recommended input level. So for example, on the farthest left, the Spark has been compared against supplier K, which recommends at least 10 nanograms of input. And we can see across the board that we get higher yields, and even at those lower input levels, all the way down to 1 nanogram. Of course, with FFPE, again, this is a highly degraded sample type that's very challenging to work with. And again, across the board, even at 10 nanograms, which is our lowest recommended input for FFPE RNA, we still see the highest yields possible and very sufficient yields for Illumina sequencing. Overall, this shows good results even with difficult samples, generating sufficient library for sequencing, meaning that you can test samples that may previously have been unable to test with other kits. Now, this is also some interesting data. So similar to our DNA-seq kits, our RNA-seq kits also show improved uniformity and better overall coverage uniformity. So this again is looking at FFPE RNA with limited input quantity. And ideally, we should see a nice flat line with a steep drop on the extremes. And this is looking at a normalized transcript length. What we can see here is the Spark in light blue with 10 nanograms FFPE RNA input, as well as a 25 nanograms FFPE RNA input in the light green. And we see a curve that we should expect. And this indicates overall the coverage uniformity is very uniform across the length, as we should hope for. And especially when compared to other kits, we're seeing high coverage across the length. And this allows you to ensure that you're getting correct identification of full length of genes. So instead of only being able to look at small portions of transcripts, the overall coverage uniformity allows us to look at the full length and to get better analysis. So even with this limited input, low quality sample, we're seeing overall very, very good uniformity of coverage, indicating good libraries in and good data quality coming out. As I mentioned, there are several options for looking at RNA transcriptomes. And the other option that we have for library preparation is our Spark mRNA-seq for gene expression analysis. As I showed you before, again, there's still the three main methods. The mRNA-seq kit utilizes poly-A selection. And this makes it great for samples that may not be mammalian. So anything that's not mammalian, such as plants, can go into a poly-A selection kit because it's not specific for the species. The challenge here is that it does require higher quality RNA because that ensures that the poly-A tail is intact and that allows for that poly-A selection to take place efficiently. But this opens this up to any researcher, say, who may be in the agricultural field or maybe working with animal species that are non-mammalian. Overall, this is a very, very similar workflow to the one that I just presented, so I'm not going to go through every step individually. But again, here you start with total RNA, and the main difference between the two kits is that initial choice between depletion and mRNA isolation. So with the
Emily, I think the sound is muted. Can you hear us? Hi, Emily, can you hear us?
Yes. Hello. Can you hear me?
Something technical happens. Let's see how can we get back on track.
Okay.
Yes. Okay. Now we are on again.
Okay, we're still live.
Yes, we are live. We are live.
Great. Then I will resume where I was at.
Sure.
Thank you, everybody, for your patience during some technical difficulties as we sometimes encounter in this field. So just to continue, I am currently discussing our Spark mRNA-seq kit, which utilizes poly-A selection from highly intact, high-quality samples. So again, in comparison to our Spark RNA-seq kit for HMR, which utilizes depletion, the only difference is really in the initial steps. So this kit takes as input total RNA and goes into an mRNA isolation module, which utilizes oligo-DT bound beads for the mRNA isolation. From there, the protocol is very similar to the HMR kit that I have already discussed. The only difference is that first reaction, instead of including depletion, only features fragmentation and random priming, which can be anywhere between 3 and 10 minutes. So very, very short. From there, directly into tube S in the same tube, as a continuation of the addition of sequential reagents. So into that same tube after fragmentation and random priming is the first strand synthesis reaction, and then add in the third set of reagents for second strand synthesis. Basically, the rest of the protocol is the same from there on. The total time, again, is about 4.75 hours. So very simple, very easy to use, very limited hands-on time for simple manipulation. Similarly to the other kit I presented, still very important is the efficiency of the mRNA capture. So here we can see we're looking across the full input ranges. So we recommend an input range between 5 nanograms up to 1 microgram of RNA. And we can see here we get very high quality and very consistent exonic coverage and very, very minimal reads dedicated to ribosomal RNA. So this indicates very efficient poly-mRNA isolation, as well as very consistent and reproducible mRNA isolation. From there, we can also look at the library yields. And what we've done is we've compared against some of our main competitors on the market and their lowest recommended input. And as we can see here, we can see consistently higher yields, as well as in some cases, higher yields with fewer PCR cycles. And this is true even down to our very, very low sample input levels, indicating again that even with low inputs, you should have more than sufficient yields to go directly into library sequencing. Finally, again, a very important metric is to ensure that you're getting reproducible and reliable transcript profiling. So what we've done here is we've looked at 50 nanograms in duplicates, and here we see very, very high concordance in the transcript profiling, and that indicates high reproducibility from sample to sample. Finally, I'd like to introduce some of the supporting products that are available separately that are compatible for both of our DNA workflows as well as our RNA workflows. First, I would like to start with our Spark Pure Mag beads, which are a magnetic bead for PCR cleanup and size selection. This is a single product solution, so this has been demonstrated to have equivalent performance to 3 commonly used products in this area. So to start, we see very equivalent DNA cleanup and size selection compared to Ampere XP. As you can see here on the bottom left, using the exact same ratios, we see the exact same profile. So this is a great drop-in solution that can save you money and see very equivalent performance to what you're currently using for library preparation. We've also compared this to SprySelect, which is often used for size selection. And here again, we see high concordance between the two products with identical profiles for size selection. So very easy to use, very easy to evaluate against what you may currently be using in the lab. And finally, this has also been shown to be equivalent to RNA Clean XP, which allows you to use for RNA cleanup as well. So this is a very versatile product, works very reliably for all of these 3 options, and is also actually room temperature stable for up to 6 months. So particularly for anybody who may be familiar with library preparation and doing bead cleanup, typically you have to wait for your beads to get up to room temperature. In this case, we have demonstrated stability for 6 months at room temperature, so you can just take the beads off the shelf and go directly into your library prep, saving you time as well as money. And then finally, another accessory product I'd like to discuss would be our Spark Universal Library Quant Kit. And this is for quantification of Illumina libraries prior to sequencing. So even in this day and age, qPCR library quantification is still considered to be the gold standard for reliable and accurate quantification of NGS library prep. And the reason is because you utilize primers that are specific for the P5 and P7 ends of the library, ensuring that what you're measuring by qPCR is exactly what would be able to ligate onto the flow cell and be sequenced. So this is going to give you the most reliable and quantification method, and will also give you superior run-to-run uniformity with these highly precise measurements. Now, we've adapted this kit to be significantly faster than others on the market. So the cycling time is only about 40 minutes compared to traditional kits which are about 80 minutes. And we've also designed this to be highly stabilized and ready to use. So historically with other kits, you have to create a master mix, you have to add in several reagents. For this kit, the Spark Universal Library Quantification Kit, the master mix is already ready to go with the primers added and the is at the correct concentration, so that you can take exactly 16 microliters of the master mix and then either 4 microliters of sample or 4 microliters of standard. So significantly reduce pipetting steps so that you save time on the manual side as well. And what we've done is we've compared this to one of the head competitors on the market, and we see here very consistent concentration quantification for different library prep types, different sample inputs. And this ensures that, you're getting very good run uniformity with much easier handling and speedier results. One thing I would also like to mention as well, so we also, as a support of Citogem, we are with QUANTABIO, and we are happy to provide any support you may need for your library preparation or other steps associated with that. We also do have video tutorials available for on-demand support, and this is available for all of our kits. And this is a really great resource because it goes through everything you may need, shows you directly how the libraries look from a hands-on perspective, and just makes it significantly easier for reference as you tackle these library preparation. So thank you very much for your time. We have a great portfolio here at QUANTABIO provided by Citogem, and we are happy to support all of the applications shown here, not just next generation sequencing, but we are very happy to help you make your lab life easier. Thank you so much.